Okay, thank you, Gordon, for that kind introduction. It's also worth just saying that Marjory Stephenson was the first ever woman to be admitted to the Royal Society as a fellow in 1945, after 300 years of that being an all-male institution. Okay, so I was absolutely thrilled to receive this award. As a final year PhD student, I gave my first ever talk in public at a Microbiology Society meeting in Sheffield in 1984. So my connections to this society go all the way back to the very beginning of my career. And just as an aside, I can remember being very nervous when I gave that talk, and I came off stage and headed for a friend of mine looking for some kind of reassurance that I'd given a halfway decent talk. And the first thing he said to me was, "Why did you give your talk in a Birmingham accent?" So don't get me wrong, I mean, I love a Brummie accent, but I'm from Croydon. So Howard's remark was extremely disturbing and not at all the reassurance that I was looking for. Okay, so I'm not going to attempt to give any kind of overview of my whole career, but rather I'm going to focus on a central question that we addressed in the 10 years or so before I retired. And that question concerns how this molecule, cyclic di-GMP, which is one of the most important intracellular signaling molecules in bacteria, controls progression through the life cycle of this organism, Streptomyces venezuelae, or SVEN to its friends. So obviously I need to introduce two things, so I'm going to start with SVEN. So as you may have heard if you happen to be in one of the sessions on Monday afternoon, Streptomyces are filamentous gram-positive soil bacteria, and they have quite a complicated life cycle for a bacterium. They distribute themselves, they disperse themselves through sporulation, and when a spore germinates, it sends out a germ tube which grows by tip extension and by branching, and that gives rise to a ramifying mycelial mat underneath the agar, which is called the vegetative mycelium. And when the vegetative mycelium runs out of nutrients, it sends out specialized reproductive structures called aerial hyphae or reproductive hyphae, and they grow out of the aqueous environment of the vegetative mycelium into the air. And they also grow by tip extension, but they don't branch. And eventually they arrest tip extension and they undergo a massive synchronous sporulation septation event. So this is probably the most spectacular moment in the life cycle. They lay down this long ladder of sporulation septa, and these eventually mature into pigmented spores, long chains of pigmented spores. So one of the great things about doing genetics on this organism is that you can identify mutants in this life cycle without going anywhere near a microscope. So mutants that are blocked at this stage that can form the vegetative mycelium in the normal way but can't form the reproductive structures look like this. So you're looking at something shiny. This is the surface of the vegetative mycelium, and these things are called bald mutants for obvious reasons. And so so these usually are defined by mutations in transcription factors. So if you hear me talk about bald transcription factors, they're required for the formation of the reproductive structures. And then mutants which can form the reproductive structures in the normal way look like this. So they acquire the fuzzy appearance of the wild-type colony because they've made the reproductive structures, but they remain white on prolonged incubation on plates. And that's because one of the last steps in spore maturation is the deposition of a polyketide spore pigment, which in the case of SVEN is this pale green color. Okay, so also just to say that superimposed on this life cycle, Streptomyces produce an enormous array of secondary metabolites, including something like 60% of the world's commercially and clinically important antibiotics. And those two things, antibiotic production and the life cycle, are tightly coordinated, both temporally but also genetically. Okay. So I'm just going to show you a small clip from a movie made by Susan Schlimpert, who's here in the audience, covering this moment in the life cycle, probably the most spectacular moment in the life cycle. So Susan makes these movies in a microfluidics device on an inverted epifluorescence microscope. And she grows them in the microfluidics device and then induces sporulation by flushing through nutrient-depleted spent medium. So what you're going to see is two reproductive hyphae growing down from the top of the slide. And Susan's introduced an FDSZ YPET fusion so you can visualize where the sporulation septa are going to form. So as they sporulate, there's a massive upregulation of FDSZ expression, and that FDSZ resolves into these beautiful ladders of FDSZ rings. And each of these rings will then recruit the cell division machinery and ultimately drive sporulation septation at each of these positions. So this event separates the reproductive hyphae, which is a single cell that might contain, say, 50 copies of the chromosome, into 50 cells, each of which contains one copy of the chromosome. So in a way that I'll explain in a few minutes, in 2012, we discovered that this molecule somehow controls progression through this life cycle. And cyclic di-GMP is one of the most important second messengers in bacteria. And before our work, it had mostly been studied in gram-negative bacteria, where it controls the so-called stick or swim lifestyle choice. So in gram-negative bacteria, low levels of intracellular cyclic di-GMP promote planktonic swimming, and high levels of cyclic di-GMP promote biofilm formation. But very little was known about what happens in gram-positive bacteria. And then in 2012, Natalia Tschowri joined the lab as a postdoc, and she did an extremely simple but very telling experiment. So Natalia simply engineered either very high or very low levels of cyclic di-GMP in Streptomyces venezuelae. And when she engineered very high levels of cyclic di-GMP, she got colonies that looked like this under the electron microscope. So this is a classic bald phenotype. So high levels of cyclic di-GMP permanently trapped Streptomyces in vegetative growth and prevented the cells entering reproductive growth and producing the aerial hyphae. So that was a really striking result, but it was actually the opposite experiment when Natalia engineered very low levels of cyclic di-GMP that gave an even more striking result, because when she did that, she got minute colonies. And those colonies were really soft and gooey, so very much unlike a classical fibrous Streptomyces colony. And when she looked under the electron microscope, it became apparent why they were like that, because those tiny colonies consisted entirely of spores. So the entire biomass had sporulated. So low levels of cyclic di-GMP caused the organism to race through its life cycle and precociously hypersporulate. So in one sense, this was a very simple experiment, but it profoundly changed our view of what it was we were working on. And as a consequence, inevitably, our focus shifted to trying to understand how cyclic di-GMP might control progression around this life cycle. And given the dramatic effects of engineering cyclic di-GMP levels in Streptomyces, it seemed that it implied that this signaling molecule must somehow be interacting directly with the regulatory cascade that controls progression through development. And that's exactly what we've been working on for the previous 10 years. So unfortunately, you can't identify cyclic di-GMP binding proteins bioinformatically. You have to identify them the hard way. You have to identify them experimentally. And so to do that, we took advantage of a compound sold by a company at that time called Caprotec, who are based in Berlin. And this molecule is effectively cyclic di-GMP on a sort of fishing line. So at the other end is biotin, so that you can recover this molecule on streptavidin-coated beads. So what Natalia did was she grew Streptomyces venezuelae in liquid sporulation medium. She took samples at regular intervals. She made extracts from each of those samples, and then she threw in this so-called capture compound and then recovered that capture compound back on streptavidin-coated beads, washed the beads, and then gave them to the proteomics facility of the Institute and asked them to identify any proteins that were bound to the beads. And when she did that experiment, she hit the jackpot because the most abundant protein on those beads was a protein that we knew very well. And that protein was called BALD. And I hope that the designation BALD will tell you that this is a protein, it's actually a transcription factor, a repressor, that this protein is required for formation of the reproductive structures. So Natalia, BALD isn't just any old transcription factor. BALD is the master repressor of the entire regulatory cascade. So I know that everybody likes to call their favorite protein the master repressor or the master regulator, but in this case, it's really justified. So the details of this regulatory cascade are not important in this talk. All you need to know is that BALD sits right at the top of it. And BALD's job is to prevent the cells from entering sporulation. So its job is to tell the cells to keep growing vegetatively. And BALD controls a huge regulon of nearly 200 sporulation genes, and it sits on the promoters of all of those genes and keeps them shut off during vegetative growth. And amongst that regulon are the genes that encode nearly all the components of this regulatory cascade. So all of these red lines indicate genes that, or indicate proteins that are encoded by genes that are directly repressed by BALD. So BALD shuts off the regulatory cascade during vegetative growth, but not just the regulatory cascade, it also, for example, shuts off the expression of FDSZ. So FDSZ is not really required during vegetative growth in Streptomyces, and as you saw, it's massively upregulated when sporulation occurs. So we had identified all of the targets of BALD in vivo using ChIP-seq. So Natalia took some of the promoters that we'd identified as direct targets of BALD and did simple in vitro EMSAs or band shift experiments and rapidly showed that BALD was only able to bind DNA in the presence of the nucleotide. So in the absence of nucleotide, so BALD is present at constant level in all of these channels, and as she titrates in cyclic di-GMP, you can see it induces BALD to bind to its target sequence. And Natalia then went on to take apart BALD. So BALD has two domains. It has a well-recognized N-terminal DNA binding domain, which with an obvious helix-turn-helix motif, but it had a C-terminal domain, which was of unknown function. And Natalia showed that this unclaimed real estate in the protein, the C-terminal domain, was a cyclic di-GMP binding domain. And furthermore, she showed that in the absence of nucleotide, BALD was a monomer, but that when you added nucleotide, it caused the C-terminal domain to dimerize. And as a consequence, it caused the whole of the BALD transcription factor to dimerize. And BALD has to be a dimer to bind to the inverted repeats in its target promoters. So at this point, we had this rather naive model that there'd be somewhere in the C-terminal domain, there would be a ligand binding pocket where the nucleotide would sit, and that when that happened, it would cause some kind of conformational change that would allow the C-terminal domain, and hence the whole of BALD, to dimerize. So at this point, we contacted our structural biology collaborator, Maria Schumacher, at Duke University in North Carolina, and asked her if she could try to solve the structure, not of the whole of BALD, but just the C-terminal domain dimer bound to cyclic di-GMP. And when Maria solved that structure, it turned out to be a remarkable structure in multiple ways. So this is that structure. So here's one C-terminal domain shown in blue and one shown in pink. And the first huge surprise was that there was no ligand binding pocket in the C-terminal domain. The second surprise was that although this was a tight dimer, there were no protein-protein contacts in the dimer. Instead, the two monomers in this tight dimer were held together not by one molecule of cyclic di-GMP, but by 4 molecules of cyclic di-GMP. And they're color-coded in this slide, one shown in green, one shown in blue, one in red, and one in yellow. So each monomer folds over on itself, and then it intercalates into a second molecule. So you have 2 side-by-side intercalated dimers, which also form lots of hydrogen bonds to form this tetrameric cage. And I'm hoping that this will now rotate. Yes, just to show you that a little bit more clearly. So ball D is dimerized by this tetrameric cage of cyclic di-GMP, which acts as an interdomain linker to drive dimerization of the protein. And Maria went on to solve multiple structures of this protein, bound to nucleotide, bound to DNA, and so on and so forth. And this is a composite structure derived from that work. So here you see the C-terminal domain held together by cyclic di-GMP, and that allows the N-terminal domains to come together and bind to the inverted repeat in all 200 or so sporulation genes that are repressed by ball D during vegetative growth. So it's not ball D per se that represses all of these sporulation genes during vegetative growth, but rather the complex of ball D bound to cyclic di-GMP. So when Natalia went back to Germany and started her own lab, she measured cyclic di-GMP's levels across the life cycle. And what she found was that they're high during vegetative growth, and they drop progressively, reaching a minimum around the transition to sporulation, and then cyclic di-GMP levels come back up again as the cells sporulate. And of course, the first and the last in this two are essentially identical, because this is spores and this is spores at the beginning here. So what we think happens is that when cyclic di-GMP levels are high, ball D is an active dimer and it's bound to DNA and it shuts off the sporulation genes. And when cyclic di-GMP levels drop, ball D dissociates and comes off DNA, leading to activation of the sporulation cascade and formation of the reproductive structures. So through this work, we were able to show that cyclic di-GMP controls the formation, the first major transition in the life cycle, the formation of the aerial hyphae, by controlling the activity of the ball D master repressor. But this, of course, raised the obvious question: did cyclic di-GMP also intervene later in the life cycle? And what I'm going to show you now is that cyclic di-GMP does indeed intervene much later in the life cycle, and it directly controls the activity of a sporulation-specific sigma factor called white G. And again, I hope that the designation white tells you that this protein is not required for the formation of the reproductive structures, but rather the differentiation of the aerial hyphae into mature spores. So if you knock out white G, you don't get spores, and as a consequence, you get white colonies that are devoid of spore pigment. But Kelly Gallagher, when she joined the lab as a postdoc, showed that if you overexpress white G, you get normally sized but very odd-looking colonies like this. And again, when she looked under the electron microscope, it became obvious why, because these colonies again consisted of an enormous heap of spores. So overexpressing white G causes hypersporulation. Okay, so this is like Goldilocks and the Three Bears: too much, too little, and just right. Okay. So the first really exciting result that Kelly got when she joined the lab is summarized on this slide. So what Kelly did was she grew Streptomyces venezuelae in sporulation medium. Again, she took samples at regular intervals throughout the life cycle. But in this case, she split each sample in half. And with one half, she made protein, and with the other half, she made RNA. With the protein samples, she did a Western blot using white G-specific antibody. When she did that, we got a big surprise, because First of all, white G was abundantly present during vegetative growth, and white G is a sporulation-specific sigma factor, so we were not expecting that. And in addition, white G levels were completely constant going into the onset of sporulation. So that was unexpected. So with the RNA samples, Kelly assayed transcript levels for a direct target of white G. So white G directly activates another gene called white I. The details of that don't matter. But Kelly assayed white I transcript levels as a proxy for white G activity. And when she did that, she saw that in the same 5-hour period that white G protein levels were completely constant, white G activity went up 15-fold. So obviously, there are lots of potential explanations for this kind of disconnect between protein levels and activity. But our prejudice, and it was really no more than that, was that this was likely to reflect some kind of post-translational regulation. And given that white G is a sigma factor, it raised the obvious possibility that perhaps white G was regulated by a cognate anti-sigma factor that we didn't know about. So to address that question, we made 2 enormous sonicated DNA genomic libraries in the bacterial 2 hybrid system pioneered by Daniel Ledon and his colleagues at Pasteur in France. And then when these libraries were constructed, Kelly probed them using white G as bait. And satisfyingly, when she did that, she got lots of dark blue colonies. And when she sequenced those colonies, most of them contained in-frame fusions to the same previously unknown open reading frame, so open reading frame of unknown function. And because this was a sonicated library, she also looked at the endpoints of those clones, and each one, because it was unamplified as well, each clone had a different endpoint, but was an in-frame fusion to the same open reading frame. And so Kelly called that gene RSIG for regulator of sigma white G, and you'll see why that name is justified in just a moment. So this gave us real confidence that the product of that gene was interacting directly with white G. And to demonstrate or to find out if that was true, Kelly co-overexpressed RSIG and white G in E. coli in the same cell at the same time. And when she did that, when she broke the cells open and ran the extracts down a nickel column, the 2 proteins came off in what looked very much, they came off together, and they came off in what looked very much like a one-to-one stoichiometry, even though only the sigma factor was his-tagged. So following this, Kelly did some simple genetics, the most striking results from which were simply the deletion of the RSIG gene. So when Kelly deleted RSIG, she got a hypersporulation phenotype, which was essentially indistinguishable from the phenotype she saw when she overexpressed white G. So overexpressing the sigma factor or deleting RSIG gave the same hypersporulation phenotype. And all of these results, both biochemical and genetic, were consistent with the idea that RSIG was a white G-specific anti-sigma factor, which we had not previously known about. And just as a small sidebar, I'd like to say that I think the reason it took us so long to find RSIG is that in most anti-sigma sigma systems, the 2 genes are genetically adjacent and co-transcribed in an operon. But in the case of our 2 genes, the sigma factor gene and the anti-sigma factor gene are separated by a full 1,500 kilobases on the 8 megabase Streptomyces venezuelae genome. So they're not genetically linked. And then the other thing is that RSIG is a unique anti-sigma factor. It's not homologous to any previously identified anti-sigma factor from other systems. Okay, so whenever you have a sigma anti-sigma factor system, something has to control either the association of the 2 proteins or the dissociation of the 2 proteins to release the activity of the sigma factor at the appropriate moment in that system. So this raised the question, what controls the activity of RSIG in order to release white G at the right moment in the life cycle? And obviously we had a strong prejudice as to what that might be, but in fact we got a definitive answer to that question much, much more quickly than we could ever have imagined, because we sent the complex to Maria Schumacher at Duke, and in record time, Maria managed to grow crystals of this complex that diffracted to almost 2 angstroms. And when she solved the structure of that complex, sure enough, buried in the middle of the complex were 2 molecules of cyclic di-GMP. So I just want to show you how cyclic di-GMP is bound in this complex. So the 2 molecules are partially intercalated, and one is shown in yellow and one is shown in green. And these 2 molecules of cyclic di-GMP sit on the 2 helices of an antiparallel chord coil. So in this representation, the first helix runs from right to left, and it contains an ESRQD motif, and these 5 residues make key contacts to the nucleotide. The second helix comes back in the opposite direction, and it contains the exact same ESRQD motif. So you have 10 residues that make entirely symmetrical contacts to an entirely symmetrical cyclic di-GMP dimer. And these 2 helices are linked by a long meandering loop that comes up over the top of the nucleotide and then goes down behind in this representation, in this projection, comes round and is linked to the N-terminus of the second helix. And we think that cyclic di-GMP acts as a kind of chaperone in this system because to order and fold this loop. And that's critical because this loop forms the major interface with the sigma factor, which sits behind the anti-sigma factor in this projection. So if I just go back one slide, it was obvious that almost all of the contacts to the nucleotide were with the anti-sigma factor. So this raised the obvious question, can the anti-sigma factor bind nucleotide by itself? And Neil Holmes in the lab and Maria Schumacher together showed that yes, indeed, the anti-sigma factor binds cyclic di-GMP tightly in the absence of the anti-sigma factor. And the second question that we wanted to answer was obviously the most important one, which was, is the nucleotide required for complex formation between the 2 proteins? And Neil and Maria showed, yes, indeed, that first of all, that RSIG alone binds the cyclic di-GMP, and that RSIG must first bind nucleotide before it can complex with the sigma factor. So in other words, nucleotide controls complex formation. So the way we like to think of this is that RSIG, the anti-sigma factor, comes off the ribosome unarmed, and then it's armed by 2 molecules of cyclic di-GMP, and it can then bind to white G and sequester it in this inactive complex. So when cyclic di-GMP levels are high, RSIG sequesters white G, and late sporulation genes are turned off. And when cyclic di-GMP levels drop, RSIG releases white G. It can then bind to core RNA polymerase and form white G holoenzyme and activate late sporulation. So in this way, we were able to show that cyclic di-GMP controls both of the major developmental transitions in the life cycle. It controls the formation of the reproductive structures by controlling the activity of bald D, and it regulates the differentiation of the reproductive structures into mature spores by controlling the activity of RSIG white G. So during sporulation or during development, bald D has to come off DNA before RSIG releases white G. And the most simple explanation for that order of events, the most we think revolves around the KDs of these proteins for the nucleotide. So the KD of bald D for cyclic di-GMP is 2.5 micromolar, whereas the KD of RSIG white G for cyclic di-GMP is 0.4 micromolar. And that means as cyclic di-GMP levels decrease, bald D will become inactive before the anti-sigma factor RSIG becomes inactive. And that leaves one really huge question, which of course is how cyclic di-GMP levels are controlled across the life cycle. And what I haven't told you is that there are 10 enzymes in Streptomyces venezuelae that can either synthesize or degrade, make or break cyclic di-GMP. And in this diagram, the biosynthetic domain is shown in red, and the degradative domain is shown in either blue or green. So you can see of these 10 enzymes, 5 of them have both the ability to synthesize and to degrade the nucleotide. And almost all of these proteins are multi-domain proteins with GAF, PAS, and PAC signaling domains attached to them. So presumably whatever signals these domains respond to control the activity of the enzyme and even potentially the ability to switch between synthesis and degradation. So obviously a major question for the future is how this ensemble of enzymes comes together to control changes in cyclic di-GMP across the life cycle. And don't forget that this is a multicellular organism, so we're not just talking about temporal changes in cyclic di-GMP, but also spatial changes in cyclic di-GMP within this multicellular colony. Okay, so I'm going to stop talking science there because I very much want to thank a lot of people. So I've been extremely fortunate throughout my career to have a succession of extremely highly motivated and very talented PhD students, postdocs, and research assistants. And so I'd very much like to thank everyone on this slide who, of course, did all the work that we've published over the years. I'd especially like to thank 3 people who acted as long-term research assistants in the lab. And I'm reluctant to use the word research assistants because these people are actually highly innovative scientists. So Maureen Bibb ran the lab for almost 20 years. And amongst the myriad of things that Maureen achieved in her career, she was almost single-handedly responsible for creating all the tools and resources that we needed to adopt Streptomyces venezuelae as a new model system for the genus. And the reason for that was that the system we worked on before, the classical model species, Streptomyces cedicolor, which is a wonderful organism in many ways, had a major restriction from our own particular interest, which is that it only sporulates on agar plates and will not sporulate in liquid culture, which was a major drawback for us. And so Maureen spent almost 3 years of her life creating all the tools and resources we needed to adopt Streptomyces venezuelae, because Streptomyces venezuelae sporulates in liquid culture, and switching into this new model system was a game-changing thing for us, for which I will be eternally grateful to Maureen. Maureen was followed by Matt Bush, who was with me for 8 years. Matt produced an endless series of beautiful experiments and published an endless series of fantastic first author papers and reviews. And then when Matt left the lab in 2018, we were in a situation where we had several very interesting projects which had big holes in them, and I was extremely fortunate that Neil Holmes joined the lab for those last 3 years and did all of the experimental work to complete those papers and get them across the finishing line. And as it happens today, today is Neil's 40th birthday, and he's in the audience, so happy birthday, Neil. I'd also like to thank a whole array of absolutely amazing collaborators. So hopefully I don't need to explain why Maria and Dick have been so important to our collaboration. You've seen that they provided detailed mechanistic insights into some of the systems that we worked on. And it's very hard for me to see how we could have got those insights in any other way. And they collaborated on many other projects which I have not talked about today. And then in the last 3 years, we established another fantastic collaboration with a cryo-EM structural biology lab run by Liz Campbell and her postdoc, Mira Lilic. And all of these people, all of these structural biologists were not only, I mean, first and foremost, they were fabulous scientists, but also they were really generous and great fun to work with. So I'm hugely grateful to them. I collaborated with Klas Flaird, a cell biologist in Sweden, for almost 25 years, and my collaboration and friendship with Kloss has been a rich part of my career. And then close to home at the John Innes Centre, Govind Chandra, the bioinformatician in the department, and Kim Finney, a superb electron microscopist. Govind and Kim have been ever present as co-authors on our papers, and it's really, we simply couldn't have succeeded without their talents. So I'm extremely grateful to them as well. I couldn't possibly give this seminar without mentioning these three scientists. So in the early part of my career as an independent group leader, I learned a huge amount from David Hopwood, Keith Chater, and Mervyn Bibb. And David, Keith, and Mervyn taught me a huge amount, most especially the power of genetics, but also the need to make sure that any biochemistry I did connected to important biology in the organism. And those were hugely valuable lessons. I'd like to thank Paul Hoskisson and Matt Hutchings, who actually nominated me for this award. I don't think the toad was involved, but thank you to him as well. And I'd like very much to thank Susan Schlimpert and Tung Lei, who have provided me with a home as an emeritus fellow in the department since I retired. And finally, I'd very much like to thank you for coming and for your attention. Thank you.